Hello everyone. For decades, scientists have believed that humans are born with fixed brain cells, i.e. fixed neuron cells. And once these cells get damaged, they cannot be replaced with new neurons and cannot go to cell division. But recent discoveries prove this belief wrong. So what is brain cell regeneration? What are the historical perspectives and recent discoveries behind it? And how does it affect human brain health? So let's find out in this video. Brain cell regeneration or neurogenesis is a process where new neurons are formed in the brain. This happens in the hippocampus region and subventricular zone of our brain. Neurogenesis is seen mostly in adults. It decreases with age but does not stop. Most 20th century neuroscientists observed that neurons are different from other body cells, that they cannot undergo cell division. Once neurons mature, they appear to be in a permanent state of rest, known as the G0 phase of the cell cycle. At that time, microscopes and imaging technologies were rare. Due to this, neuroscientists could not find much evidence of neurogenesis. In the beginning of the 20th century, father of neuroscience, Santiago Ramon gave this statement for the human brain that in adult centers, the nerve paths are something fixed and it immutable. Everything may die, nothing may be regenerated. At that time, this concept had become dominant. Because of this, findings of other researchers were ignored, such as in 1960s, Joseph Altman and Gopal Das provided evidence of adult neurogenesis in rats, although their findings were rejected by the scientific community. But in 1990s, researchers like Elizabeth Gould and Fred Gage provided solid evidence of neurogenesis in adult mammals. And recent research such as in 2018 by Maura Boldrini and their colleagues found evidence of neurogenesis in the hippocampus of the human brain and suggested that neurogenesis is lifelong, which breaks the old concept of neuroscience and proves that brain cells can be regenerated. In adults, neurogenesis is divided into 4 stages. Number 1. Proliferation. In this phase, neural stem cells divide and form progenitor cells. Neural stem cells are multipotential cells that can generate all cell types of the adult central nervous system. Number 2. Differentiation. Progenitor cells now undergo differentiation and they become immature neurons. Differentiation is a crucial step where the neuron cells transform from the progenitor stage into a specialized neuronal state. Number 3. Migration. In this stage, immature neurons migrate from their birthplace to their final destination. This migration ensures that new neurons or immature neurons arrive at the correct location where they are needed to drive neurogenesis. Number 4. Integration. In this phase, migrated neurons form connection with existing neurons. Integration involves establishing synaptic connections that allow new neurons to communicate with existing neurons. It contributes to brain functions like other neurons by being a part of the brain's neural circuits and contributes to cognitive processes such as memory and learning. Begins to function as new neuron cells. Neurogenesis plays an important role in memory and learning. Its reduction has been linked to conditions such as depression and anxiety. Research has shown that promoting neurogenesis helps in the treatment of diseases like Alzheimer's and Parkinson's. Positive influences like exercise, diet, learning, and meditation can increase it, while stress, aging, and substance abuse can reduce the rate of neurogenesis. So this was today's video. How did you like this video? And if you like it, then please subscribe to us.